These are the steps of meiosis. It first starts with interphase, which is where the DNA replicates and the protein synthesis occurs. The next step is called prophase. This is when the DNA condenses, causing classic chromosome structures to appear. Once they've appeared, metaphase is next. The chromosomes then align on the spindle fibers in the middle of the cell. Next is anaphase. This is where the chromosomes separate onto two different sides of the cell. Then there's telophase. This is when the nuclear membrane appears around each set of chromosomes and they're still attached. The next step is cytokinesis. This is when the two cells form, causing two daughter cells. After cytokinesis, the phases then repeat, starting with interphase 2. This time it has three parts. It has the G1 phase, the S phase, and G2. In G1, RNA is synthesized, in the S phase DNA replicates, and in G2 the cell grows more. Then in prophase 2, the chromosomes in the two daughter cells prior pair up. Next is metaphase 2. This is when the prior chromosomes line up in the middle of the cell. The next step is anaphase 2. This is where the sister chromatids pull apart and go to separate sides of the cell. Next is telophase 2. This is where the cells pinch in the middle and start to form 4 cells instead of 2. The last step of meiosis is cytokinesis. This is where the nuclear membrane finally comes off and the 4 new daughter cells are formed. In contrast to mitosis, in meiosis, 4 new unique daughter cells are formed instead of 2 identical daughter cells. These are the steps to meiosis.